So, welcome everyone. Welcome to the OTS webinar series. My name is Ronald Buijsen, and I'm co-hosting today's webinar together with Jillian. And for today, we will focus on the OTS poster winners of the OTS 2022 meeting. And we have two poster winners that will present their data today. So we have Irene Vázquez Domínguez and we have Paolo Pigini. I let the opportunity to the presenters to introduce themselves. So Irene, the floor is yours.
Hi, good afternoon, everyone. I'm Irene Vázquez. I'm a postdoc researcher in Radboud UMC, and today I will present you my research about chemical modifications in the retina. So I hope that you like it. And I think that I can share the screen now, right? Or
yeah, go ahead.
You can share the screen. Yeah, go ahead.
Okay. Yes, so I think that you can see the presentation mode now, right? Okay. So as I mentioned, today I want to share my research that is assessing the efficacy and safety of delivery of different chemically modified antisense oligonucleotides to the eye. So in our group, we investigate inherited retinal disease or IRDs that are caused by the progressive degeneration of this tissue that is highlighted here in yellow in the inner part of the eye, that is the retina. And it consists in different layers that, when worked properly, allow us to see our environment. However, that means that when one of these layers is disrupted or affected, they can lead to visual impairment. Just to mention that around 50% of the mutations that cause inherited retinal disease are associated with effect in splicing. And that's why we use antisense oligonucleotides as a promising therapeutic tool. But all you know, because we are in the OTS, that these molecules are not RNA naked molecules. Usually they present chemical modifications that improve their efficacy and their stability. In this talk, I will mainly focus in these three modifications: the two OME phosphatidylides, the two MOE phosphatidylides, and the PMOs. But just to simplify the talk, I just refer them just as OME, 2 MOE, and PMO. So I just told you these modifications improve the efficacy and stability, but we don't know how they can affect the uptake, biodistribution, and toxicity in the retina. So for that reason, no comparison has been made so far. So for that reason, the aim of our research is to conduct for the first time this comparison in order to assess what are the effects of these chemistries in the uptake, biodistribution, and toxicity in a retinal context to improve the current therapies. To do that, we select two genes that are expressed exclusively in different retinal cell types. We have the photoreceptor, rods and cones, the bipolar cells, and the RPE. In the first part of our research that was conducted in vitro, what we did is to introduce this gene entirely or partially in MIDI genes or space report vector, and we also designed AONs divisible by 3 in order to have a very clear readout. So we designed the AONs in order to cause the skipping of the target exon, as you can see in this graph. So we designed different AON sequences per MIDI gene, and we first compared them just harboring one of the chemistries. And for that experiment, we select the sequence that worked better in modulating splicing. And with that sequence, we compare the 3 different modifications at RNA level to assess what worked better or worse in splicing. So I want to start briefly, just mention what we obtained in these in vitro results. And I want to start with a rhodopsin. That is the most representative results. So in this case, we were able to clone the entire gene of interest in our MIDI gene system, and we select the AON1 that is able to target and cause the skipping of the exon 4. And we assess this splicing with primers located in the exon 2 and 5. So in this electrophoresis picture, you can see all the conditions that we have. We have the not treated cells and the untreated MIDI gene. So the first conclusion that we can assess is that the MIDI gene works because we have an increment in this band that represents the wild type transcript from this point to this point. However, when we transfer the 3 different modified AONs, as you can see here, we reduce these wild-type bands and we observe an increment of those bands we are skipping alone, in which the exon 4, is skipped alone or together with the exon 3. In addition to these modified EONs, we also have our schema oligonucleotide. There is a random sequence that cannot interact with the desired pre-messenger RNA and therefore works as a negative control. And if we compare all the replicates that we have, we clearly observe that there are no differences between our schema oligonucleotide or our treated condition, indicating that the effects that we are observing in this yield are sequence but not chemistry specific. But if we compare the three modified EONs, the 2-OME, the 2-MO, and the PMO, clearly the 2-MO results to be the most efficacious one in modulated splicing. However, we are in science and we are not always the things, or in our case, the mitigene works equally, and we have some exceptions. And I want to tell you about GMCs that will be with us along all my talk. In this case, the gene is quite long to be cloned entirely in the mitigene system, so we use an alternative. We just introduced part of the gene of interest that we wanted, in this case, from the intron 5 of GM6 to the intron 9, and we introduced a mitigene between the human rhodopsin exon 3 and 5. And it is because this exon 3 has an alternative transcription start site that allows the expression of this region of interest. In this case, the EON that we selected, the EON 3, target and cause the skipping of the exon 8. So as I did before with rhodopsin, we can go through the data. And the first thing that you probably notice is that exon is naturally skipping our mitigene system. So we have the wild-type band here and the skipping of the exon 8. However, when we transfer the different modified EONs, we observe an increment of the skipping of this section and a reduction of the wild type. But if we compare the three chemistries, there are no differences between them since that all of them were more or less the same independently of the chemistry. But as I mentioned, it's an exception in a 5 of 8 mitigenes, the 2-MO modification results to be the most efficacious one in splicing modulation. And the PMO modification on the other side results to be the less efficacious one. But at that point, we thought that probably it's because the naturally chart of the PMO that difficult a lot to work with that chemistry in vitro. So with this in mind, we went to the second part of the study that was the in vivo. In this case, we decided to inject the different modified EONs in the right eye of the mice, while in the opposite eye, we inject the same amount of PBS. And it's that way, each animal is on control. 1 week later, we harvest the retinas to proceed with the RNA analysis to assess splicing as we did in vitro, but also we collect some ACAPs to conduct morphological studies and assess potential effect in toxicity. So I will start with the splicing modulation, and I want to come back to GM6. If you remember, I just told you that in our mitigene system, we don't see differences between chemistries. However, here in the upper part, you have a representative animal per modification, and we compare the untreated eye versus the treated eye. And it's clearly, you probably observe that not the three chemistries work the same. And as you see below the graph, in which I have all the replicates of all the animals, we clearly observe that the 2-MO is the most efficacious in increasing this band of here that correspond with the skipping of the exon 8 and reduce the wild-type band. But again, the sun doesn't show any differences, indicating that these effects are sequence but not chemistry specific. So in that point, for these experiments, we conclude that the 2-MO also works better in vivo. So now I want to move to the morphological studies and just mention that we conduct the entire comparison of all chemistries in one gene per retinal cell types that are the ones that I highlighted in blue, rhodopsin, NAC2, GM6, and RDS5, plus the SON, the scrambled oligonucleotide. And one thing that you need to remember always that work with animals is that you need to follow up the animals after your procedure, because you need to ensure there are no side effects. So with that, we follow the animal up to 74 hours. And what we observe is that in 88.5 of the animals injected with the PMO modification, they present this ocular phenotype that you see that the eye is looking like white. And this appearance and this effect was not detected in the opposite eye of the same animal, neither in the other chemistries. So if we look further what is happening here, we clearly observe that mostly of the animals present a dilatation of the pupil that is highlighted in blue, or even cataracts. And as I mentioned, that happened in all the conditions, even the scrambled oligonucleotide. This, together with the fact that the retinas that we had were whiter and smaller, make us to think that probably this is a PMO-mediated toxic effect. So to assess what is happening in this PMO condition, we first decided to see the overall structure of the retina. And we did that by doing a tinction by toluidine blue. So I will start mentioning what is happening with the animals injected with the EONs, with the 2-OME modification against NACTU, so exactly these animals of here. And here you have the untreated eye, the one injected with PBS, and the treated eye with the EON with the modification. And I think that you can see that there are no big difference between them, right? The same tinction, the same size, more or less the same structure, so they look apparently the same. If we go to the 2-MOE modification, we have the same staining again, the same size, so okay, more or less the same. But when we went with the PMO, we clearly observe differences. For one side, they have some points of the retina that seems to be thinner, but more importantly and more evidently is that there is a detachment of the retinal layer from this black line of here that correspond to the choroid and the RPE. So in order to validate that is happening in all the eyes or not, we assess the same in the remaining PMO condition that you have here. And in all the cases, we observe detachment. In some cases are bigger than in the others. And the retina, also we observe that is quite thinner in some points. And also like in the case of the RDS5, we see that the staining layer that we observe here in the treated eye is not the same that we observe in the PMO. So our next question is like, how are the different retinal layers affected for this? And for that, we're conducting some immunohistochemical studies. But before going there, I want to go back briefly to this slide. And it's just to highlight there are 2 points in the retina, in the bipolar cell layer and the photoreceptor layer. There is a big accumulation of the nucleus of these cells that can be easily visible by DAPI staining. And these layers are known like inner nuclear layer and outer nuclear layer. And if you are doubting what is what, the bigger one always corresponds to the photoreceptors. So with this in mind, I want to move to the immunohistochemical studies. And I will start with the EONs again, rhodopsin. So we have here one representative animals, untreated and treated eye with the 2-OME modification. And the same here, we have a representative animal with the untreated eye and the corresponding opposite eye treated with the 2-MOE. So we have in blue the DAPI staining that I just mentioned you. In green, we have the GFAP, that is a stress marker that is highlighted in green. In purple, you have rhodopsin that mark the outer segment, so the area that is just located here on top, in this case, on top of the outer nuclear layer. And also PNA in red that mark the areas surrounding the cones and also in other areas. So we compare in both cases the untreated and the treated eye. We clearly observe there are no marked differences. The retina looks practically the same. However, again, if we move the PMO, we see some differences. In this case, if you observe the staining of the outer segment and the outer nuclear layer that is clearly visible in the untreated eye, is totally lost here. So we don't observe this layer anymore, and we have like a co-staining of rhodopsin, PNA, and DAPI. And also we have an increment of the GFAP staining in green, indicating that these cells are quite stressed. So as we did with the toluidine blue staining, we wanted to validate this result in the remaining PMO condition. So I will start with the PMO against RDS5 and the scrambled oligonucleotide. And in this case, as happened with the antisense oligonucleotide array against rhodopsin, we lose totally the X structure. We don't understand any more outer segment, outer nuclear layer, and even here the inner nuclear layer is almost invisible. This effect is also detected in the sun in this area of here, in which we observe a completely loss of the structure. If we go to NACTU, well, I think that no further explanation is needed. The picture is quite clear. There are no layers at all. We have just a mass of cells with co-staining of all the markers that we're using. But when we move to GM6, we surprisingly observe that we have again all the layers. But if you focus on the outer nuclear layer, this one is to be much smaller than in the opposite side. So we decided to measure this value, the value of the outer nuclear layer, not only in this size, so in all the condition and eye when possible. And after statistical studies, we observed there is a significant reduction of this layer after the PMO injection. So all this data together support our initial hypothesis that this is a PMO-mediated toxic effect. But for those that are familiar with the in vivo morpholinos, you know that they are not alone. Usually for in vivo, this morpholino oligo present a coadjuvant that is injecting with them. In our case, is this moiety that is here in black is an octaguanidine dendrimer that you can see is quite big, that is linked to the morpholino oligo that is here represented in green. So that means that in any injection that we did in the animal, we are actually injecting two compounds, the morpholino and the coadjuvant, and any of these compounds could be responsible for the toxicity that we are observing. So in order to know if the moiety is the responsible itself, we decided to remove this part of the equation by injecting the modified PMOs that we use in our in vitro studies. So the first thing that we noticed, and we're super glad to show that, is that the mice look super good. They have no ocular phenotype anymore. Also, the retinas that we harvest are equal in size and color between the untreated and treated eye. We didn't detect any differences in toluidine blue staining, neither in the immunohistochemical studies. So there is no toxicity here. However, is this unmodified PMO still able to redirect splicing as we desire? So for doing that, we repeat this analysis that you are now familiar with. Here we have two representative animals treated with the unmodified PMO against GM6 and unmodified zone. And again, we observe that in the treated animals, we have an increment of this band of here in the skipping of the exon 8 that was not detected before. And also the screen oligonucleotide didn't show differences, so it was great. However, if we compare the efficacy in between the unmodified PMO and the in vivo PMO, we clearly observe that this one, the modified PMO was less efficacious than in vivo. So we are wondering that maybe the uptake can be affected by the loss of this moiety. So in order to assess what is happening in uptake, we are now assessing a new technique in the lab that is the microRNA scope. And I'm doing together with my colleague Dia, and we are using the Biotechnic kit. So this is a summary of how this kit works if you are not familiar with. First, we need to permeabilize our tissue, in this case, our retinas. Then we design a proof against our AONs, in this case, the GMCs that we employed. Then we use different reactive to amplify the signal to be able to visualize them. And we see like red dots in the area that is joined the proof with our AON of interest. So in the last month, we are trying to optimize first the technique for the positive and negative proof that comes with the kit. The negative proof apparently cannot join to anything, and the positive proof is joins to a ubiquitin expressed RNA. And it's the result that we have. So we also again use the DAPI staining that I mentioned you before because it's very easy to combine the proof that is in red and the merge of the two channels. So we clearly see that the negative control have no any staining in the retinal layers that you have represented here. But if we move to the positive control, we observe a positive staining in the ganglion cells, the inner nuclear layer that is here, the outer nuclear layer, and also in the RP that are located here. So one that is assessed, we decide to give a try with our proof against GM6. And for that, I take three material for the three retinas with the three different chemistries that I use in the first batch of animals. So the 2-OME, the 2-MOE, and the in vivo PMO. And this is the result that we obtain. So we compare the staining. We observe that in the 2-OME, we have a positive staining like the positive control that we use with a clear tinction in the ganglion cells, the inner nuclear layer, the outer nuclear layer, and the RPE. Sorry, in the other way. Here we have the tinction in the ganglion layer, and here we have improved the outer and the inner nuclear layer. And you can see that the staining of the outer nuclear layer that is here, that corresponds to this part, is a little bit more weak than in the other. And also we have a staining, also quite weak in the RPE. And we observe a similar result in the PMO. So as I mentioned, there is just a try, is a test. We are still trying to optimize the result because probably you see that it's quite deposit of the proof in our samples a bit. So we are still trying to optimize, but at least seems that our proof is able to recognize our AON in our samples. So when we have all the optimization ready with this proof, the next step is to validate these results and then to compare the uptake between the unmodified and the in vivo PMO to assess if the uptake is affected, we thought, And for that reason, the splicing is the spacing is different. So as conclusion, just mention and just remember that the two MOE modification were super good, even in vivo and in vitro. That regarding the AON efficiency, we obtain similar results in vitro and in vivo, and even some AONs showing higher efficiency independent of the chemistry in vivo. That neither the two MOE or two OME modification result in toxical effects in the mouse retina, but as I mentioned to you, the in vivo modification result in a clear toxic retinal phenotype. And taking into account the last result that we obtained in vivo with the modified PMO and so on, that points that probably the octaguanidine dendrimer is the responsible for this toxicity, as has also reported in other tissues. With that, I want to conclude you. Thank you, for your attention, the organizer for giving me the opportunity to present here today, and I will be happy to answer your questions.
Great, thank you so much for that, Irene. That was a fantastic talk and so remarkable to see all the data that you've been able to generate. We'll now open it up to the Q&A, so feel free to anyone in the audience to use the Q&A function in the webinar, and we're happy to get those started. While people are thinking of their chat and their questions, I'm happy to start off with a question. I guess my first question is, do you have an understanding of where that toxicity may be coming from that you've been observing?
In the octaguanidine dendrimer?
Yeah.
You mentioned. Well, we don't know for sure because we need to do tests of what's happening in the retina, but we are not the first one that we observed this. And it's a paper that mentioned that when they inject this PMO, also with the octaguanidine dendrimer modification, they have like a precipitation. And also with the tissue is quite irrigated, they generate coagulation that leads to the death of the tissue. So maybe it's that part in the retina too, but we need to validate that now. For now, it's only an hypothesis. We know that we are not the first one, so we are happy that we are not us. But also it's not clear how the moiety can affect. Is the problem with this in vivo morpholino? There is not a lot of information just about the moiety itself, so it's quite complicated. But seems that in some tissues with a lot of blood vessels go in that way.
Yeah, that's very interesting, especially with the retina. It's such a vascular organ in a way that it's, there's just so many variables, especially in such a tight space.
Oh, yeah. Can I ask a question? Because so I think the eye is considered as a kind of a closed compartment, right? Mm-hmm. And I think the same is true for the brain. But when you do ICV injections, direct injections in the brain, I think there is a lot of leakiness. So I think the majority of the ASOs just goes to the liver and to the kidney. So is there any information when you inject these ASOs in the eye of animals where the majority of the ASO goes? So does the majority stay within the eye, or does it go to liver,
theoretically
kidney?
They stay there for the retinal blood barrier. We also think in the past, Alex and I think, to conduct a study to validate that, but at the end we don't get the money. But apparently all the references and all the reviews saying that the retinal blood barrier keep the AONs there. So we don't expect that a big amount of AON goes somewhere else. And also the amount that we injected is quite small because it's a very small area too. So the dose, even if they go through somewhere, if we lose a big part, we didn't see any effect in the eye, and we see effect. So probably stay there the majority.
Yeah, and related, no, not a related question, but another question is, so I think what you now observe is chemistry-related toxicity due to the PMO chemistry. I was just wondering, so are there like any tools that you use to analyze general toxicity? So are you doing any RNA sequencing studies or any other studies to assess toxicity? Because it's not always observed, right? Because you inject it in the eye and then probably you collect the eyes after a few days, weeks, months.
One week.
Any ideas? Yeah, any ideas?
No, for general toxicity, like measure stuff in the liver or stuff like this, we are not doing yet, but we harvest the material. So we harvest kidneys, liver, and brains to assess potential toxicity also in the system of the animal.
Great, thank you so much. We have a few questions in the chat. So we have questions saying, I once have seen relationship between TM and toxicity. How are the thermal stability of those ASOs?
Well, for the AON design, we use different tools, and all the AONs are quite stable. So all the parameters, length, size, thermal sensitivity, and everything are very equal between them. And they are reported to be stable. I don't remember the number by heart now, but we assess that are quite stable.
Great,
and as I mentioned, we have the same sequence with the different chemistries, some of the toxicity. Yeah, in that sequence, not in all of them.
Wonderful. Another question is, could injection trauma cause cataracts as well?
Well, we do an intravitreal injection, so we deliver the AON in the space between before the retina. So usually, if the injection goes well, usually the animal have no effect. So we are also surprised to see cataract. Usually we see cataract like not related with the injection. It's just something that happened animal per se, but not for the injection itself. Usually with this injection, we have a lot of expertise and we don't have any cataracts until now.
Okay, that's very interesting, the relationship there about what may be driving that. So next up question we have is, dear Irene, nice presentation. Related to Ronald's question, could you track the AON fluorescent label?
We thought about that, but the problem is that with you on a fluorescent compound, the uptake can change because the AON becomes to be much bigger. So we are not sure how the fluorescence can affect the result, and especially if this is comparable with the sequence that we are using. So for that reason, we decided to go to the RNA scope, because at the end is the sequence that we inject with no any other tag. I mean, with the in vivo PMO, we already have the moiety, so imagine to add also another thing on top. So in that way, we ensure that it's nothing related with the label that we include, and it's just everything related with the AON that we are using.
That's very interesting. And I feel like that technique overall will be a fascinating, just once all these ASOs and AONs are inside organs, it's always so complicated to track them. So I feel like now that at least having a bit more of a visual could be a huge asset.
Yeah.
Yeah, I think John Watts published a technique where you can determine the concentrations of the ASO within your tissue of interest. So that might also be something to apply. And I think nowadays there are also some ASO backbone antibodies available. So you can collect your tissue of interest and do an antibody staining and see whether this stays positive for the ASO. That might also be something to take into account.
Yeah, I will consider that too. Thanks.
Wonderful. Thank you so much. So just once again, thank you so much for speaking with us today, Irene. And we'll now pass it off to our second speaker, Paolo. So Paolo is a postdoctoral research fellow at the Agency for Science, Technology and Research. So Paolo, if you'd like to quickly introduce yourself and then get started to share your slides.
Yeah, thank you very much for your introduction, and thank you for having me here. Now you should be able to see my presentation, I think. So yeah,
yes,
my name is Paolo Pigini, and I work in the Institute of Molecular and Cell Biology in Singapore in David Weiss lab. And today, actually, my story is quite similar or parallel to Irene's stories, but I wish to have all those nice data. So I'll try to play with what I have, way more humble. So basically, we apply these engineered small nuclear RNAs in different contexts, pathological contexts. But in this case, I'll be talking about an application in another kind of retina disorder. So this is my disclosure slide. And well, basically, in our lab, we use different tools to play with splicing, because splicing, as you all know, is a highly regulated mechanism, and it's also quite modular for each axon. So you can use, as you all know, different tools to modulate it. Like you can knock down a target gene with antisense oligonucleotides by inducing exon skipping and a frame shift. This happens, for example, in Huntington disease, or in Duchenne muscular dystrophy. You can do the same thing, but to restore exon the protein frame in case there is a frame shift mutation. And you can do the same thing with basically not only with antisense oligonucleotides, but also with modified small nuclear RNAs. And at the same time, you can use antisense oligonucleotides to basically restore exon skipping, where this has been caused by mutations by splicing silencers that actually induce exon skipping, and you can use antisense oligonucleotides to restore normal splicing. But the case I'll be talking today is about these donor splice site mutations that induce exon skipping and can be restored with this engineered small nuclear RNA called U1. So the U1 is a small nuclear RNA that we all have in our cells. It's quite important for splicing because it has this antisense sequence that is complementary to the conserved motif of donor splice sites. And its function is to bind to donor splice sites and trigger splicing by interacting with the spliceosome machinery. But mutations occurring in this binding region can prevent this and induce exon skipping. So the very simple idea is to reintroduce an U1 with an adapted antisense sequence adapted to the mutation and restore splicing. Now, if you go through like databases like CleanVar that collects annotated pathogenic variants, you try to look at the location of these variants in the transcript. You can see that there is an abundance of these annotated pathogenic variants in proximity of donor splice sites. This is in part due to positions +1 and +2 that are very important for splicing, but all the other mutations are likely to induce the pathology because they affect the binding of the U1. And there are more than 1,000 of these variants that are very likely to induce the pathology because of this process. And that's what we're trying to address in our project. We basically developed our own platform to deliver these modified U1s. It's just a plasmid that expresses the U1, and you can customize the sequence to adapt it to any mutation. And you can deliver through lentivirus, AAV, but for therapy, AAV is preferred, of course. And we're trying to test it on different conditions, but today I'll be talking about our lead case study, which is about this gene ABCA4, which is associated, its mutations are associated to Stargardt disease. So just last introduction slide about this disease, Stargardt, which affects the retina, the macula actually, and more specifically the photoreceptors. So normally in the normal visual cycles, you have light that is absorbed by these photoactive compounds, like in this case retinol, 11-cis retinol, which is converted into all-trans retinol. And all-trans retinol in photoreceptors is shipped to RPE cells, retinal pigment epithelium cells, to be converted. I very simplified the process, but I'm not going into details. But basically, it's shipped back to photoreceptors where it can be used again for the visual cycle. And ABCA4 protein is a membrane protein that has a very important role in this process. And the loss of ABCA4 induces an accumulation, leads to an accumulation of toxic compounds like this N-retinidine P, also lipofuscin in RPE cells, and this causes cell death. So there are more than 1,000 ABCA4 variants, and when they are in this biallelic recessive condition, they can induce cell death in the retina. The typical onset age is around 20s, but can be variable. And after a few years, some retinal lesions become larger and larger until the patient gets blind. So it's quite an impairing disease. So our idea is just to deliver the U1, mutation adapted U1 to the retina through AAV, and try to rescue the phenotype, try to prevent this degeneration. So our next step was to look for mutations that could be fixed with a U1 therapy. This is ABCA4 locus. The cDNA is quite large, so it cannot be really, it's not really suitable for gene replacement. So you need something like ASOs, the U1 or CRISPR. And we found some mutations that can be potentially addressed with the U1 technology, because they induce exon skipping, frame shift, and eventually you lose the C-term part of the protein, which has a catalytic function. So the mutation, our lead mutation is, I will not disclose it today because of we're trying to file a patent about this, but it's not really important, the coordinates, just I call it +5 GA, because it's in position +5 from a donor splice site. So basically, this is the mutation and the affected exon. And the problem of ABCA4 is that it's not expressing basically any cell line, only in photoreceptors. So in order to do some first in vitro tests, we had to develop a mini gene, not a mini gene like in the previous presentation. So it's a bit smaller. It has just a fragment. It expresses a fragment of the gene with exons surrounding the mutation, and it's supposed to replicate the splicing environment around the mutated exon. So the idea is to co-transfect the mini gene and the U1 to a model cell line like 293T. And as you can see from this variant-specific PCR, the wild type mini gene creates a splicing variant with the exon. Instead, the mutant mini gene is affected by this exon skipping, so it loses exon X. And if you reintroduce the normal U1 that we all have with a normal sequence, you don't have any rescue. And the curious thing is that if you reintroduce the U1 with a single adaptation of the mutation, you don't have any rescue as well. But if you introduce a U1 with full complementarity to the mutation, and it's full complementary because the endogenous U1 is not fully complementary, it's just partially complementary. So if you reintroduce a U1 with full complementarity, you are basically capable of rescuing splicing, as you can see also from this quantitative PCR. And I'll talk about later why this single adaptation is not enough, even though it's counterintuitive. Just keep it in mind for now. And yeah, if you introduce full complementary, you basically can rescue splicing, but rescue is not, as you can see here, 100%. So the next step was to try to improve this efficiency. And we basically exploited a native mechanism of the U1. We designed a walk of U1s, not just targeting the canonical U1 binding sites. And they were all supposed to work because there is an innate mechanism called splice site selection from a distance that allows the U1 to work from a distance from the canonical site. So it's quite recent. In the last 5 years, there has been the evidence that the U1 can work not just in the canonical site. And this allows us to make a walk of U1s and try to select an optimal one in case it works more efficiently than others. So we design our walk for our mini gene. And as you can see here, some U1s targeting some specific positions shows higher efficiency, as you can see here. So basically, we could find some optimal U1s in terms of efficiency. And very importantly, as you can see from this variant-specific PCR, there is no splicing aberration induced by the U1. And I mention this because in other cases, the U1 can induce some unwanted splicing aberrations. Like in this case, this is a different mutation from a different exons that we're working on. And we also designed a walk. And as you can see here, when you introduce different U1s, you have these aberrations that are indeed intra-retention, which are not there for the wild type condition. So the U1 can also induce something negative. So it's something you have to deal with when you try to design an optimal therapeutic tool. And I try also to keep this in mind because I will try to put everything together in the final slides. But for now, it's important to, the important message is that you also have to take care not only of efficiency, but also about safety. So our next step was to deal with off-targets. Because the U1, the antisense sequence is short, is 11 nucleotide. It can be longer, but usually 11 nucleotides, and it targets conserved motifs. So potentially it has off-targets. And we, as we did before, we can exploit this selection, this bulk of U1s, trying to select U1s that have a minimal number of off-targets, of predicted off-targets. So now we're talking about prediction. So it's not that simple because the U1 is known to tolerate alternative annealing registers and mismatches. As you can see here, it can tolerate bulges, double bulges on both strands and asymmetric loops, and it's known to tolerate mismatches. So the potential targetome is quite large. And we developed on our own a pipeline to predict off-targets based on these known dynamic features that basically takes the input sequence, defines the targetome, the potential targetome, and blasts the targetome through a specific dataset, in our case, the human transcripts. And we validated our pipeline on the endogenous U1, which has some known targets, which are basically all the donor splice sites of the human genome. And as you can see, the pipeline can find a high frequency of targets at donor splice sites, even if you decrease the number of annealed bases. And we applied this pipeline to predict the off-targets of our bulk of U1s. So these are the all fully complementary off-targets without considering any mismatch or alternative register. And as you can see, some U1s have less predicted off-targets. These are off-targets all over exons and introns. But then we also looked at off-targets at donor splice sites or acceptor splice sites, because these are very sensitive, could be very sensitive. And if you, as you can see, there are some U1s that might be optimal, and the situation doesn't really change if you introduce in the pipeline mismatches and alternative registers. So basically you can define optimal U1s in terms of minimal of targets, and they partially overlap with the efficient U1s. So in this way, we could identify optimal candidates for therapy. And after this initial screening in vitro, actually, we're all working in vitro. We don't have mouse models, but we try to move to a model which is more similar to retina. And the problem of ABCA4 is that it works in a very specific cellular environment, which is the cooperation between photoreceptors and RPE cells. So this is kind of a work in progress, and I don't have full data about this. I just want to mention that we're trying to differentiate photoreceptor precursors and RPE cells and organoids to try to see, first of all, splicing and rescue of ABCA4 in these models, and also try to do some functional tests, even though it's not that simple. And for now, we just work with RPE cells, but other data are coming. And if we reintroduce the U1 in the mutated system, we can rescue splicing of the transcript, and we can also rescue the protein. Also, the protein is not simple to work with because it's not very expressed. And the next step will be to do some functional tests and try to assess the real off targets through RNA sequencing. And just a few more slides in which I'm trying to put together some things about the mechanism of the U1. So as I mentioned before, reintroducing a single modification to restore normal splicing, in this case, seems not to be enough, even though it's counterintuitive. You need full complementarity. And we actually thought that this was a problem about structure, because you can see here that the U1, the endogenous U1, has this structure. This is just a prediction model. While if you try to predict the structure of these other two U1s with different modified antisense sequences, you can see that this single modification by chance can affect the secondary structure, which is quite important for the U1 to work. While always by chance, this combination of nucleotides of bases in this antisense sequence does not affect the structure of the U1. And this is just prediction, of course. And so the structure is one thing to take care of when we design the U1. The other thing is that the U1 can induce some unwanted aberration. And in this case, we thought that the problem could be to the strength of the U1 binding, because the U1 is supposed to interact with the spliceosome to trigger splicing. But at some point during splicing, the U1 has to be displaced. So if the binding is too strong, this could be a problem, and you might have intron retention. So we're trying now to put everything together, try to correlate efficiency with different variables. And I try to make it more easy to read. And as you can see here in this dispersion chart, every dot represents a U1 from a different exon, from a different mutation. And this is efficiency. As you can see, the red ones, the red dots, have high efficiency in rescuing some mutations. The blue dots have low efficiency. With the same chart, with the same U1s, if you look at the GC content of the antisense sequence, which represents, in a way, the strength of binding, you can see that there is a correlation between low GC content and high efficiency. And this could explain why, if you have high GC content, high binding strength, you might have aberrations and low efficiency in rescuing. At the same time, if you look at another factor like the distance from the canonical splice site, you don't really see any correlation. But it's important to mention that here, all the distances are from a range that is supposed to work. So it's not too far. It's close enough to work, but the very close distance from the canonical splice site is not really an issue. So you can basically design your U1 at any position. At the same time, if you look at the structure, and this value represent the distance between the structure of the U1 and the structure and the correct structure, so the lower the better, there is kind of a correlation between efficiency and correct structure. So last thing, trying to put everything together. If you have a mutation that induces a 5' splice site mutation that induces exon skipping, and you want to design a U1, and the U1 has bad folding, or it's way too far from the splicing, you're not going to have any rescue. If the splice site, the target site is in place, and the structure is correct, you can rescue splicing. But at the same time, if binding is too strong, you might have aberrations like intron retention. So all these factors together, we're trying to put them together in our current projects, just to provide some guidelines for an improved design. And with this, I just want to mention that all this work is also mainly thanks to big collaborations between different labs in IMCB. So it's a big effort, not only for this project, also for other projects. And but I'm grateful to all of them. And thank you all for your time and for this opportunity to be here. So I would be happy to take any question for now.
Yes, Paolo, thank you very much for this excellent presentation. I really enjoyed it. So I think a very obvious question, maybe the first question from my side. So I think in the previous presentation from Irene, she showed that different chemistries might have like an impact on toxicity. So did you ever compare these U1s with ASOs of different kinds of chemical modifications? And did you ever test these similar ASOs in your model to do the same?
Actually, we're working also with ASOs with different kinds of mutations, trying to, for example, restore splicing. And toxicity is indeed an issue. I think Irene's data are very interesting. We're also working with Morfolino. So I think she has way better data than us, because we're not working with mice. And so we cannot really provide that kind of information. But from the U1 perspective, it's kind of different because the U1 is delivered through AAV, so it's expressed internally. So if you exclude any toxicity due to AAV, which should not be there because we are thinking to use a carrier that is already FDA approved, should be fine. If you exclude that, the U1 per se is known not to have big toxicities, unless the antisense sequence you use has off targets, which is what we are trying to understand. So the U1 in general seems to be safer than antisenses because it has no chemistries, but that's something we need to demonstrate.
Yeah, no, that perfectly makes sense. So we have a question in the chat. It's a question from Alex Garanto. Thanks for the presentation and the overview on the use of U1. My question is, it is reported that mini genes can lead to wrong splicing patterns, and that in fact, the larger the context, the better. Have you evaluated the effect in cells or in a bigger context for all the variants? Perhaps I missed it, but how do you deliver the U1s? And for how long is the treatment? And last but not least, how recurrent are the variants you are studying, and how do you see the translation to the clinic? So these are a lot of questions in one sentence.
So
the context.
Yeah, the first question was about the mini gene, and it's absolutely true. The mini gene is very artificial and can lead to very weird stuff. And that's why probably I was too fast here, but we basically trying to see if we can rescue splicing in the same way also in retina cells, not just with the real ABCA4. This was not a mini gene. So yeah, we're trying to do that. Apparently in this case, it's efficient, but in other mutation we try to deal with, also in ABCA4, also in the same systems. The mini gene works, so we can rescue splicing in the mini gene, but we cannot rescue splicing in the real ABCA4, in the full length ABCA4. So yeah, the mini gene is artificial, is useful, I think, when you want to screen a large number of U1s that you cannot do like if you do more complicated experiment. And once you define your optimal U1s, which we found that they're still optimal also in the retina models, then you can work with that. Basically, so the mini gene is more useful for screening.
Yeah, I agree. That's very, especially for these external walks, it's super feasible. Maybe to add one question to this. Did you ever block nonsense mediated decay, just to see what kind of different splice forms you have within your different models?
Yeah, absolutely. So all the PCR that I showed, they are actually treated with CHX to block nonsense mediated decay. Yeah, that's very important because sometimes you might trigger something and you might not see it because it goes through NMD, but it might do something as a real biological effect. So yeah, that's something we do by standard.
Let's see. So the delivery aspect. How do you deliver the U1 to RPE?
Yeah, the AAV. Basically, there is this FDA-approved AAV, which is a serotype 2, pseudotype 7 or 2, I don't remember, which is used to deliver RHO genes. RPE65 to another kind of retinopathy. So it seems to work. There are better AAVs for the retina that have been proved in literature, but this is the only one to be FDA approved. So our idea to go through to the clinic is try to use that kind of AAV to deliver the U1, and it seems to work. We're already working with that.
Oh, great. So how long is the treatment?
Yeah, so in theory, the retina cells are not supposed to replicate that fast. So the AAV does not integrate; it stays in the nucleus as an episome. But so it could get lost in tissues like the liver, right? Because you have a new liver every, I don't know, 3-4 years, something like that. But in the retina, it should be kind of stable. So always, also in theory, just a few injections should be long lasting, a few years. But actually, honestly, I don't know if there are data about the other AAV FDA-approved AAV. That would be very interesting to see if you need more injections over your lifespan. But in theory, less injections than an ASO, which has a different kind of delivery problems. I'm not trying to bring a fight between ASOs and U1s, of course.
No, but it's always good to make a comparison. You have your pros and cons from every strategy and from every model. So it's always good to highlight the advantages, but also the disadvantages from every technique. So I think it's always good to do.
I would say that in general, okay, we're doing different things, but ASOs are very efficient, very efficient. Instead, the U1, you have to really try to push the efficiency up to 100%, but ASO are very efficient.
Yeah. So then the question, so how do you see the translation to the clinic? Kind of relates to this.
Yes, it's basically we need first of all to prove that we can do our tricks also in real retina cell lines, and then the let's say the killer experiment that we don't have in mind right now is to work with the mouse with AAV, with the FDA-approved AAV. And we actually in Singapore we have a patient that who has a mutation that we can rescue with a U1. So that would be our gateway to the clinic. But unfortunately, she has some other mutations also in ABCA4, so it's a bit complicated. But yeah, we're trying to address different kind of mutations. Our best leads, we will try to push them to the clinics by testing eventually in mouse with AAV.
Yes, and then this is about your prediction. So have you ever correlated your prediction of target genes with a real experiment?
That's we're actually trying to do that. We have tried in a small scale, like with targeted PCRs, and the U1s that I'm showing in this presentation, they have overall very low off targets, but there are some U1s that have many off targets, and if you try to check the off targets by PCR, you can see the effect. The major effect is rescuing, forcing in exons that are by nature are spliced out. So some exons are very weak splice sites, and the U1 can force them in. So that would be an off-target effect. And we're just now we're waiting for the retina models to be ready, just to do an RNA-seq and to check for our U1s to have no off targets. But yes, we proved that the predicted off targets can have a splicing effect and eventually a biological effect.
Yeah, it's always good to do like a combine, right? To check the predicted off target effect, but also to do it in an unbiased way. Yeah, good. So a lot of questions from Alex. I would assume that the AAV delivery will be producing U1 as an RNA constantly, so it will accumulate in the eye, and eventually more of target effects can appear. So that might also be a good remark. So is it like a ratio between
so apparently
U1s and generating them, and any ideas about that?
So the good thing about the U1 is that it's the most abundant small nuclear RNA in the cell. And so the cell is very used to have high levels of the U1. Our concern was that the U1 could do some kind of sponge effect and retain too many proteins, because it actually creates some ribonucleoproteins, so it interacts with protein. But apparently that's not the case. So the cells are very used to have high levels of U1s. We're also doing some titrations, trying to see how many gene copies the cell can tolerate of the AAV inside the nucleus, and apparently can tolerate them. This is about the abundance of the U1. In terms of off targets, our idea is try to select U1s that really have a minimal amount of predicted off targets. Also, because if we want to do the validation test with RNA-seq, we will do it in vitro or in the mouse. You will never do this in the real human retina. So you will never be 100% sure if there are off targets or not in the human retina. because of course you cannot take samples. That's why prediction is also very important. Try to minimize the theoretical of targets. So yeah, in terms of abundance of U1, the cell should be able to tolerate it, seems to tolerate it, also in literature. In terms of off targets, we try to select the U1s with the least number of off targets.
Yeah, thanks. And then the very last question from Dia. Nice talk, Paolo. What's your Western blot on iPSC-derived RPEs?
RPEs. I was expected this question because ABCA4 is very low expressed in RPEs, and as you can see, it's not the best Western blot ever. So we managed to see it, but it's very hard. And we also noticed that when you differentiate the cells, not all the cells are actually RPEs. Also, when you differentiate precursors, not all the cells in the plate are RPEs. So immunofluorescence is way better than Western blot, which I don't have today, but we are actually working on that.
Cool. And with that, we nicely finished within the hour. So I would like to thank Irene and Paolo for their excellent presentations. And I would like to thank Jillian for co-hosting today. So it's thanks a lot for helping me out. And then the webinar will be placed on the OTS website most of the time within a day. So you can always send it to your colleagues or to your friends to rewatch the video. And then I think we will have the next OTS webinar in 2 weeks. So have a nice day and see you later.
Bye. Thank you, everybody.
Thank you.